Professor_Tang_Chat_Tang_2024-10-07_18-59-44. Chat timestamp, October 7, 2024, 19:02 and 8 seconds. Professor_Tang_Chat_2024-10-07_18-59-44.txt. User, hello, Professor Grace, are you there? Assistant, hello, Professor Tang. Yes, I'm here. How can I assist you today? User, I'm wondering, are there any difference between male and female's glutathione concentration inside mitochondria? Assistant, yes, there can be sex-based differences in glutathione concentration within mitochondria, though the extent and nature of these differences depend on various factors, including tissue type, age, and physiological conditions. Potential differences. 1. Hormonal influence. Estrogen has been shown to influence glutathione levels and antioxidant defenses in cells. Studies suggest that female mitochondria may have higher glutathione concentrations, particularly in tissues sensitive to hormonal fluctuations like the liver and brain. This effect is largely attributed to estrogen's regulatory role in oxidative stress pathways. 2. Mitochondrial function and ROS production. Males and females exhibit differences in mitochondrial reactive oxygen species, ROS, production, which may influence glutathione demand and synthesis. Mitochondria in male tissues are sometimes reported to produce more ROS, leading to a higher consumption of glutathione and potentially lower steady-state concentrations. 3. Tissue specificity. The differences are not uniform across all tissues. For instance, in skeletal muscle, male mitochondria might have lower glutathione levels compared to females, while the pattern could be reversed in other tissues like the heart. 4. Age and metabolic state. Age-related changes and metabolic states, e.g., fasting, exercise, can modulate glutathione concentrations differently in males and females, adding another layer of complexity. Research context. Most of the studies exploring these differences are conducted in animal models, so human data is more limited. Understanding these variations can provide insights into why males and females might have different susceptibilities to oxidative stress-related diseases. User, can you list the titles of some research papers relevant to your previous answer? Assistant. 1. Mitochondria, a central target for sex differences in pathologies, 2017. 2. Sex differences in the phosphorylation of mitochondrial proteins, 2010. 3. Estrogens, estrogen receptors effects on cardiac and skeletal muscle mitochondria, 2017. User, does this paper, Sex-Specific Differences in Mitochondrial Function and Its Role in Health Disparities, mentioned glutathione? Assistant, the paper, Sex-Specific Differences in Mitochondrial Function and Its Role in Health Disparities, does not focus specifically on glutathione but addresses sex differences in mitochondrial function, which could indirectly involve glutathione as a key antioxidant. The paper highlights how mitochondrial function varies between males and females, particularly in metabolic pathways and susceptibility to oxidative stress, which are areas where glutathione plays an important role. Although glutathione is not the main subject, its relevance to mitochondrial health and oxidative stress makes it a potential factor in the observed sex differences in health disparities. User, what are the main points discussed in research paper titled Oxidative Stress and Aging? Is aging a cysteine deficiency syndrome? Assistant, the paper titled Oxidative Stress and Aging, Is Aging a Cysteine Deficiency Syndrome? Explores the idea that aging may be associated with a deficiency in cysteine, an amino acid crucial for the synthesis of glutathione, a key antioxidant. The paper suggests that insufficient cysteine intake could lead to impaired antioxidant defenses, contributing to oxidative stress and age-related decline in immune function, muscle strength, and insulin signaling. It proposes that dietary cysteine supplementation may alleviate some of the effects of aging by boosting glutathione levels and reducing oxidative damage. User, Can you list the key messages in the paper? Assistant. 1. Oxidative stress. Aging is associated with increased oxidative stress due to a decline in the body's antioxidant defense mechanisms, particularly glutathione levels. 2. Cysteine deficiency. A central hypothesis of the paper is that aging could be partly due to cysteine deficiency, which impairs glutathione synthesis. 3. Impact on health. The deficiency could exacerbate immune dysfunction, muscle loss, and insulin resistance during aging. 4. Dietary intervention. Supplementing cysteine may help mitigate some age-related issues by enhancing antioxidant capacity. User. Can you list some major research papers which reference this paper? Assistant. 1. Modulation of glutathione and thyroidoxin systems by calorie restriction during the aging process, 2003. 2003. 2. Free radicals and the physiological control of cell function, 2002. 3. Against the oxidative damage theory of aging. Superoxide dismutases protect against oxidative stress, but have little or no effect on lifespan in Cenerbditis elegans, 2008. User. Any other papers? Assistant. 1. Mitochondrial oxidative stress in aging and healthspan. 2. Oxidative stress in aging and chronic degenerative pathologies. These papers build on the oxidative stress framework from Cease and Droga, further exploring the role of mitochondrial dysfunction and ROS in aging-related diseases. User. Can you pack all of the content of our chat into a text file for me, such that I can go through it again later? Thanks in advance. Can you pack all of the content of our chat into a text file for me, such that I can go through it again later? Please insert a date and timestamp at the very beginning of the text file, and also add a timestamp as a file name suffix. Thanks in advance.